distinguished guests, family members, friends, colleagues and cousins of the distinguished professor in the Department of Life and Consumer Sciences in the College of Agriculture and Environmental Sciences, Professor Z. Mkhize-Kwitshana. I don't want to distinguish anyone, Professor N. Mapoli, Acting Executive Dean in the College of Agriculture and Environmental Sciences, the respondent, Professor G. Waltz, Executive Head from Stellenbosch University Immunology Research Group. Ladies and gentlemen, good afternoon. What a beautiful, warm spring afternoon to welcome Professor Mkhize-Kwitshana to the professorate when the jacaranda trees are about to blossom in the city of Tshwane, where our beautiful university, the University of the Land, is situated. I am both humbled and elevated by the honor and privilege to welcome you to the University of South Africa's virtual platform on behalf of the Chancellor, Dr. Thabo Mbeki, and the Vice-Chancellor, Professor Puleng Lengabula, on this occasion of the inaugural lecture of Professor Mkhize-Kwitshana. I'm delighted to welcome you, as I'm told that a visitor comes with 10 blessings, eat one and leave 9. So we will be left with 9 blessings. Ladies and gentlemen, this proverb is associated with a Kurdish culture and hospitality. It signifies that when a guest visits, they bring good fortune, which are blessings, with them, and even though they may partake in some of the host's resources, including eating, they leave behind a greater amount of good fortune than they consume. The proverb highlights the positive impact of welcoming guests and the reciprocal nature of hospitality. Ladies and gentlemen, my name is Mpongwepe. I'm the Acting Vice Principal, Research, Postgraduate Studies, Innovation and Commercialization. Today, I am officiating as we are witnessing and we are witnesses to the rite of passage of Professor Mkhize-Kwitshana to become a full professor. Indeed, the presentation of an inaugural lecture is a significant milestone in the academic career of a full professor, and it provides a platform to share past research and introduce new ideas to a diverse academic and non-academic audience. Hence, you are here today to witness that. This ritual has been practiced since time immemorial for hundreds of years as a way to test newly professors to profess their knowledge to a lay audience and fellow academics. Indeed, the origin of the title professor comes from the need to profess or to declare publicly one's knowledge. Today, Professor Mkhize-Kwitshana will declare her knowledge publicly on the topic from co-infections to cancer, unraveling the immune threats of neglect. Co-infection is a loaded concept. Ladies and gentlemen, it refers to a condition wherein 2 viruses invade the host simultaneously or in a short period, whereas in the case of a superinfection, the host that has previously been infected by one virus is again infected with a different virus at a later point. You can see that this is loaded. Indeed, it's an important topic at this day and age. Today we are going to learn more about co-infection that leads to cancer. We know that cancer is the second leading cause of death in the world, but survival rates are improving for many types of cancer thanks to improvements in cancer screening, treatment, and prevention. It is the works of scholars such as Professor Mkhize-Kwitshana that gives us hope. And we are told that this co-infection, it worsens human health. 76% of 2,000 publication in meta-analysis, it indicates that the infection can spread from one place in the body to the entire body through the bloodstream. But early diagnosis and treatment may prevent complications such as bacterium, sepsis, and the septic shock. These are serious life-threatening conditions that need immediate treatment. So we should relax and listen to the professor. Once again, you are welcome. Relax, enjoy the inaugural lecture and the virtual hospitality of the University of the Land. Let us be inspired so that we do not expire. Where I come from, they say, "Moeng e tla ka gae, re je ka wena." It pays to make a visitor feels at home. The proverb highlights the positive impact of welcoming guests as well. So I'm saying welcome to the University of the Land. And as we express our gratitude, we must never forget that the highest appreciation is not to utter words, but to live by them. Today, it is time to celebrate the hard work, dedication, and the significant milestone of Professor Mkhize-Kwitshana. Relax and enjoy the talk. NgesiZulu bathi izinja siboshiwe, ningakhululeka kusekhaya. I'm now going to request Professor Mapoli, the Executive Dean of the College of Agriculture and Environmental Sciences, to introduce Professor Mkhize-Kwitshana, who will then deliver the inaugural lecture, followed by the respondent, Professor G. Walls.
Professor Ngwepe, Acting Vice Principal, Research, Postgraduate Study, Innovation and Commercialization. Professor Mkhize-Kwitshana for her and her family. Professor Walzel, the respondent, distinguished guest, colleagues, and esteemed member of the academic community. Good day. Today, we are here to celebrate not only for Prof. Mkhize-Kwitshana's family and the friends, but also for our college and the university, as we congratulate her in the excellent work that she has been doing in our college and over the years in her career. Professor Mkhize-Kwitshana is currently a full professor in biomedical science at the University of South Africa. She is a C-rated medical scientist in immunologist, holding the PhD in immunology. She is registered with the HPCSA as a medical biologist scientist. She is also an honorary associate professor at the University of KwaZulu-Natal and a senior research associate at the University of Johannesburg. She holds several national and international committees position. Despite joining the academia after 14 years in public health services, where she left as a deputy head of the chemical pathology department, she established an impressive academic track records. After training and becoming the deputy head of the chemical and pathology department at the Edendale Pathology Diagnostic, she joined the SA Medical Research Council as a trainee medical scientist, where she left as a seasoned senior scientist to join the University of KwaZulu-Natal as a lecturer and research coordinator, then later promoted to senior lecturer. She was later headhunted by the Mangosuthu University of Technology to be the head of biomedical science department for 4 years, after which she rejoined the University of KwaZulu-Natal and became the associate professor. An academics manager with experience in medical research, that's who Prof. Mkhize-Kwitshana is. In higher education and diagnostic pathology, environments encompassing routine clinical diagnostic in public health sector, medical research, academic and undergraduates and postgraduate and postdoctoral teaching and supervision. She also leads and participates in several capacity development initiatives. Professor Mkhize-Kwitshana research interest is in the immunology of co-infections, with a special interest in immunological modulation of HIV and also TB and oncogenesis by neglected tropical infections. Other areas of interest includes intersections between immunity, microbiomes, infections, and nutrition. She has been a principal investigator on several research projects and has attracted millions of research grants funding over the years and has published 112 publications in these areas. She has graduated several PhDs, masters, honor students, and also mentored postdoctoral fellows. She has established collaboration nationally and internationally. She is a recipient of international and national recognition awards, including the United Kingdom Immunology International Fellowship Award in 2011, the SAMRC Scientific Merit Award for Research Capacity and Development and Transformation, and also the International Meeting Advancement Awards in 2025, the Medical Laboratory Sciences Excellent Award in 2025, and the MUT Honorary Microbiology Award in 2014, and also the Gold and Silver Research Award in 2017. Professor Mkhize-Kwitshana, she is a strategist leader, lead in including serving the 2 terms as a board member for Executive Committee member of South African Medical Research Council, serving as an ESCO and representative the board to the Parliamentary Health Portfolio Committee. Currently, she holds 2 Minister of Health appointment as a member of National Health Research Ethics Council and also a member of National Health Research Committee. She was also appointed by the DG Health to be a member of NTD Elimination Forum. She is an ex-member of South African Immunological Society. She is also a founder of the National NTD Control Task Team. Internationally, she is a guest editor for Frontiers and also in oncology, and also a guest editor in MDPI, Microorganism Special Issues, and also a editor board member for International Journal of, and also review national and international journals. Professor Mkhize-Kwitshana is a finalist in the 2025 South African Women in Science Award hosted by the Department of Science and Technology. Let's welcome Professor Mkhize-Kwitshana as she's coming to give us the inaugural lecture. Thank you.
I am Zilungile Mkhize-Kwitshana, Vice Chancellor, the Executive Management, colleagues, friends, family, esteemed guests. It is with a deep sense of gratitude and humility that I present this lecture. This moment does not belong to me alone. It belongs to all the students that I've mentored. It belongs to all the communities that have enabled the science, as well as all the institutions that have supported this work. I thank all my supervisors, I thank all the institutions, and I thank all those who are no longer here, whose sacrifice made this work possible. In particular, my late parents and my late 2 sisters. I am now going to present this work titled From Core Infections to Cancer, Unraveling the Immune Threads of Neglect, which is anchored in my core focus of immunology of core infections, looking at neglected tropical diseases as silent master regulators of other diseases. For context, the outline will comprise of the context under which the work was done in the agency, and I will report on what we have found about the neglected tropical diseases and co-infections, and then also the ongoing work and what we are planning for the future work. So with that, just a brief overview of what neglected tropical diseases are. These are selected group of neglected bacterial, viral, protozoan, and helminths or simply worm infections that are targeted by the WHO for elimination by 2030. The helminths or worm infections are highly prevalent in the tropical regions and subtropical regions of low and middle income countries, and they disproportionately affect poor communities who have no clean water supply and poor sanitation, among other poverty-related challenges. In South Africa, the two groups that are very common are schistosomiasis, commonly known as bilharzia, and the soil-transmitted helminths, in other words, those that are transmitted through the fecal-oral route, where the fecal matter contaminates the environment. In 2001, due to the sheer numbers of people being infected by these worms, the WHO recommended that all areas that are endemic for these infections must initiate mass deworming programs that are targeting school-going children. And then South Africa implemented this WHO resolution in 2016. We were about 15 years late. The challenge with this program that is targeting only school-going children leaves a critical gap, because we now know that even adults in these endemic areas are susceptible to these infections. And what is the biggest challenge is that infection with these helminth infections do not display dramatic symptoms in most of the people who are infected. In other words, many people go around without knowing that they are harboring these infections. And also, these infections overlap with other high priority diseases. Having said that, though, some of the very dramatic morbidities caused by these infections, particularly in children, as we can see here, this small intestine was removed from a 5-year-old boy who had been obviously infected over and over again, because these worms do not multiply inside the body. The amount of worms are a reflection of repeated infections. And for their transmission, a female worm of this kind of worm will excrete 200,000 eggs per day, which then get excreted through the feces, and they contaminate the environment such that more and more people get infected. Now, back to where our work was originated. In the late 1990s, developing countries were experiencing a very dramatic and aggressive HIV/AIDS epidemic compared to high-income countries. Many theories were put forward. There were propositions that the strains of HIV that were circulating in developing countries were more virulent. That was not proven. Many theories. And then it brought into focus the key difference in those countries, the developed and developing countries, where the developing countries were shown to still carry a huge burden of helminth infections, where these in the developed world have been eliminated. The hypothesis then took root. It was proposed that the chronic endemic infections with helminths were actually amplifying the HIV/AIDS epidemic. And then this was supported by epidemiological data. As we can see, those countries that were hardest hit by HIV, including South Africa, which was the epicenter of the epidemic, were also harboring high burdens of helminth infections. This is what then started the hypothesis upon which the work started. And it then brought into focus the characteristics of these worm infections. The first one being, these infections are chronic. In other words, individuals can be affected from 1 up to 25 years. These worms can survive 1 up to 25 years inside the host without them knowing that they're infected if there is no treatment. Even in the presence of treatment, in individuals who are exposed to contaminated environments, reinfection is very common. Within a few weeks or months, the treated individuals become infected. So it means that these individuals are chronically immune activated. In other words, the immune system is challenged and it's trying to get rid of these infections, but it doesn't eliminate the infections. Even though these individuals mount a very strong immune response. These worm infections have developed mechanisms to suppress and modulate this immune response so that they can survive for a long time inside the host. And one of the consequences of chronic worm infections is malnutrition, resulting in the deficiencies of all the essential micronutrients and macronutrients, which result in stunted growth, impaired cognitive development and learning abilities in children. And as we shall see, there are many other complications now associated. And then the second manifestation, because these intestinal worm infections share the same environment with our gut bacteria, the good bacteria inside the gut, they cause the disruption of the balance that is required to maintain health and a functional immune system that is caused by a balanced gut microbiome. We know that these good bacteria are acquired in utero, inside the uterus during pregnancy and during the birth process. These good bacteria actually help in the development of the immune system, and they train the immune system at infancy to be able to respond appropriately, in other words, to respond to harmful bacteria and not to respond to harmless bacteria like food antigens, and not to respond to our own body cells. So they are to be in proportional numbers all the time in order to make sure that our immune system functions properly. So with the presence of these intestinal parasites, the balance of these good bacteria is disrupted. And these have serious implications for our immune system because these type of worms will produce a specific type of immune response referred to as the type 2 immune response, which then downregulates the type 1 response that is critical for the control of intracellular bacteria such as your HIV and your TB. So if there is an upregulation of the type 2 immune response, the type 1 responses are decreased. In the presence of this chronic immune activation, the disbalanced gut bacteria, malnutrition, chronic activation of the immune system, it all then increases the susceptibility of the individuals to other unrelated pathogens. And then the chronic parasite infections also leads to chronic inflammation. The combination of all of these, the chronic immune activation, chronic inflammation, and the persistent immune suppression of by these chronic helminth infections increases the risk for the development of cancer. So in our context, because most of the surveys in the country had been done on school-going children who were known to carry the heaviest burden, our first question was, are adults in South Africa affected by intestinal parasites? And if so, what are these effects of these infections on either highly prevalent diseases that coexist with these helminth infections? Indeed, to answer this question, we undertook surveys in adult populations in the 2 tropical and subtropical provinces of the country, the Western Cape and KwaZulu-Natal. And indeed, we found that adults in these regions were highly infected. We found high prevalences of these infections, up to 60%. And not only that, we also found that up to 41% of them were also co-infected with HIV. And also importantly, when we did the socio-demographic surveys, we found that these were diseases of poverty. We found that older age, lack of sanitation, lack of clean water supply, and low income were significantly associated with these co-infections with HIV and helminths. Next, we then looked at the nutritional status of those individuals who were co-infected. And then we found a variety, all forms of malnutrition, More than 60% of the participants were obese and overweight. And when we look at the dietary patterns, in other words, the food consumed by these communities, we found that only carbohydrates were within the required dietary reference range. Other nutrients like energy, fiber, protein, and all the micronutrients, vitamins, were below. In other words, these populations' diet contained very little micronutrients and proteins. Indeed, when we also did blood analysis, we found that those with co-infections had lower vitamin A and other nutrients, to confirm what was being picked up in the dietary intake patterns. And also we find high prevalence of anemia. Even those that were not infected were almost 23% anemic. And the HIV group had the highest prevalence of anemia at 45%, followed by those with both helminth infections and HIV. Then next, we wanted to look at what happens to the immune responses of those with these dual infections. And indeed, our group, I was the first one to report in South Africa that individuals who have both helminth infections and HIV were less able to control HIV, as shown by higher viral loads, lower CD4 counts, and an impaired type 1 response, which is critical for the control of HIV. And then again, in a cohort that was recruited after the implementation of test and treat. In other words, all the participants were now on antiretroviral treatment. Even then, those who were on antiretroviral treatment, we found that there was evidence of a disarray, chronic inflammation, as shown by the pro-inflammatory markers. In other words, the type 1 immune responses were disrupted in those who were HIV and helminth co-infected, even in the presence of antiretroviral therapy. We also conducted animal models of co-infections, where we also found that the expression of immune microRNAs in mice that were infected with a helminth and the herpes simplex virus also displayed disrupted immune responses in the vaginal mucosa. We then extended our work to look at what happens when individuals are infected with HIV, with helminths and TB. Again, we found that the type 1 or pro-inflammatory immune responses that are essential for the control of TB were downregulated in those individuals who had both TB and helminth infections. And more interestingly also, the type 3 immune responses and type 2 immune responses were also upregulated, meaning that the immune suppression was there, as shown by the type 3 markers, and also the type 2 responses, which also downregulate these type 1 responses that are critical for TB control. Thus confirming in both instances TB and HIV co-occurring with helminth infections is also compromised immune responses. We then also went ahead to look at the composition of the gut bacteria or the good bacteria in those with co-infections. And indeed, we found that there is a shift in the balance in the good bacteria in the gut, as shown by a predominance of a certain types of bacteria in those with co-infection compared to those who were not infected. And then, after finding all of this, we decided to look at biological aging. Biological aging is a natural process, physiological process, that happens as we grow older. The immune system becomes less functional. However, certain conditions such as lifestyle, lack of exercise, also chronic infections will accelerate biological aging, because our immune cells, the immune cells of those who are infected are constantly multiplying. And the more the cells multiply, they reach a stage where they can no longer multiply. In other words, their function is arrested and it becomes what is called immune cynic cells. In other words, the immune cells are no longer functional. It's either they die or they accumulate. And biological aging is related to life expectancy because if we age biologically, the quality of life deteriorates and it shortens the lifespan. We measure then the telomere length, which tells us the biological aging. The shorter the telomere length, it means the shorter the faster the biological aging. Indeed, what we found was that those with the helminth infections had the shortest telomere length, suggesting an accelerated biological aging, followed by those with co-infections with helminths and HIV, compared to those who were uninfected, suggesting that these co-infections actually accelerate biological aging. Then the next question, having found all of those comorbidities that are associated with dual infection with helminths, either with HIV or TB, we then asked what are the cumulative effects of these multimorbidities in the risk for development of cancer. This very busy slides is a simplified reflection of what actually takes place in the majority of our populations who are exposed to socio-economic and environmental factors of deprivation, lifestyle factors like excessive alcohol and tobacco use, unhealthy diet, and they're also exposed to a multiplicity of infections that insult them at the same time. And these interact with their genomes that can then be affected by all of these, resulting in a whole range of morbidities that essentially even affect the immune system. Because the lifestyle diseases impact on our immune system, biological aging, in fact, impacts on our immune cells and function, a disrupted gut microbiome impacts on the immune system, and so does malnutrition because an optimally functioning immune system requires optimal nutrition. And genomic instability can also impact on the immune system. The overall effect, I will not detail all the technical terms of what happens in the immune system. The ultimate effect is that the chronic immune suppression and dysregulation combined with chronic inflammation plus tissue damage lay a good foundation for cancer development. So our focus then moved to an exposomic framework. The exposome concept describes all the total environmental exposures that an individual encounters from birth throughout lifetime and how these environmental exposures then impact on the biology and health. So we started then to ask the question, how do these cumulative life exposures and our genetic makeup, noting that the African populations have the most diverse genetic makeup, which, by the way, is not reflected. Many variants of our genomes in the African populations are not represented in the global genome. And this is the basis of the development of all the treatments that we are importing. So the question now, as we are experiencing an ever-increasing cancer burden, we are getting more and more younger people diagnosed with cancer, and our treatment outcomes are very poor. About 70% of the cancer treatments do not work. And we get more aggressive forms of the cancer. For example, colorectal cancer is characterized by increased microsatellite instabilities. So we're getting a very aggressive and a different form of cancer compared to the developing countries. And unfortunately, due to structural and systematic challenges in our country where there's limited access to care, and there's also a lack of early detection and diagnostic mechanisms, we are therefore facing a huge challenge in terms of our cancer. And the question is, could the face of our cancer be affected by all these multi-morbidities that are caused by infections, and of particular concern, those infections that are neglected, that are off the radar? So we started then to interrogate from our cohort if there are any factors that are associated with cancer. We started by looking at the markers of immune regulation and dysregulation. And then indeed we found the immune checkpoints. Those are the molecules that actually regulate the immune system. We found that during co-infection with helminths and HIV, the immune markers were actually upregulated. The downregulated or the immune suppression markers were upregulated. And one of the immune stimulating molecules was upregulated, which was a hint towards an increased risk for cancer development, because we know that a functional immune system is constantly surveying for cancer cells and it kills them, a process called immunosurveillance. So in the presence of a suppressed immune system and also an upregulated immune suppression environment, it creates a conducive environment for cancer development. We then are now proceeding to look at the different comorbidities that we have detected in these cohorts to see how they relate to the development of cancer in our local cohorts. The first work is looking at the composition of the viruses or the gut viruses in relation to the co-infection, and we want to look at what biomarkers are actually upregulated. Preliminary analysis in this study is showing that HIV and helminth co-infection viral families are related to those of the patients with colorectal cancer. And then also, we are also looking at the composition of the bacterial species that are supposed to be in balanced proportions in the context of HIV and helminth infections in relation to colorectal cancer risk, by screening for the presence of colorectal cancer biomarkers during the co-infection. And then again, we know that schistosomiasis or bilharzia affects the liver and the bladder. So we want to know whether the combination of bilharzia, HIV, and hepatitis, another liver-affecting virus, we want to know that the combination of these infections, does it increase the risk for liver cancer development? And then we're also looking for a similar increased risk for bladder cancer development in the presence of schistosomiasis and HIV. We are also then looking at the impact of environmental exposure, the microbiome composition, and co-infections, and looking for the risks for colorectal cancer development. Likewise, we are looking at lifestyle, environmental exposure, co-infection, biological aging in the context of colorectal cancer development. In the process, we are also looking for biomarkers that are appropriate and relevant for our populations which are affected by all these comorbidities. And then now, going forward, we are seeking funding to then do the whole genome sequencing so that we can then combine all the data from environmental, sociobiologic, infection, and microbiome to the biomarkers that we find to see whether then we can use the big data analytics from the big data scientists, who will then collate all this data to try then and find machine learning algorithms which will assist, for example, in cancer risk prediction and treatment predictions. This is where now our work is going, looking at the genomic and exposomic interactions so that we can then develop big data analytics which can be input for artificial intelligence and precision medicine or tailored, personalized medicine that is relevant in our context. So we are pushing towards exposomic and genomic interactions as risk factors for cancer, looking at the entire exposome and then looking at the genome and all the variants that would be associated to define the cancer risk specifically for our own local populations. This has implications for the global health and the local context. One of my passions has been capacity development, particularly individuals from historically disadvantaged institutions, and I've had the pleasurable experience of watching these, most of them young females, expanding their techniques in the laboratory and then up to a point where they get their qualifications and graduate. This slide captures one of those edifying moments where these students now reach their aspirations. However, least but not least, in the principles of the United Nations Sustainable Development Goals, which commits to leaving no one behind, I also teach the students that research must not end in theses and publications. It must also go back to the communities who have enabled them to actually develop and get their qualifications. So we do this community feedback and also even educate the healthcare providers on what our findings mean. And these are some of the moments that really complete the full cycle of research. Not only that, I also participate in this journey in advocacy. We know that there's a challenge of neglect with neglected tropical diseases. So in 2010, I co-founded the National Task Team for Parasite Control, together with the National Department of Health's Communicable Diseases Directorate, which then in 2015, I was invited to be part of the expert working group for the implementation of the National School Deworming Program that was to be implemented in 2015, together with the National Department of Health, the Department of Basic Education, and other international bodies to chart the way forward. And then in 2016, because we needed to know the distribution of these entities in the country, I was leading the grant, the emergency grant from the MRC, which was to facilitate the mapping of these neglected tropical diseases in the country. And then in 2018, I was also nominated by the DG for Health to be part of the National NTD Elimination Forum, where we are developing the national guidelines for the WHO roadmap for NTD, Agenda 2030 for the elimination of NTDs. And then currently, I am part of the technical working group on schistosomiasis and other NTDs, where we are strongly advocating for the inclusion of adults in these deworming programs for the control. So lessons for this work, we have learned that if we are to move towards AI and personalized medicine, we need to include our African exposomic and genomic data so that we can develop locally relevant prevention, diagnostic, and treatment interventions for them to be effective to improve health outcomes in our local populations. And this requires a systems approach. Instead of looking at all these morbidities in silos, we need to adopt a systems approach That will include our unique African cohorts, which will provide rich omics and clinical data to then be able to develop tailored interventions that are responsive to our local communities. And there is capacity for that because we have a strong research infrastructure in the country. And we also have possibilities with all the available bioinformatics, omics, and big data analytics. And most importantly, we need to train the next generation of scientists using this systems approach instead of the silo approach that does not work in our context. For this, we need global collaboration with partnerships in Africa and across the globe with a multidisciplinary and cross-sector approach. The one thing I have learned from this journey is that neglected tropical diseases, invisible exposures, poor communities are not fringe issues. They are core to understanding health in Africa if we are to improve health outcomes in our populations. I would like to acknowledge a range of institutions that have supported this work and also our international collaborators who have hosted a number of our students. I would like to acknowledge the MRC and the NRF for funding support, the NHLS as well for funding some of our students, the two institutions, University of KwaZulu-Natal and UNISA. And I wish to thank you all. Ngiyabonga. Thank you for your time. Thank you for your belief in science with purpose and for joining me in this journey that is far from over. Thank you.
It is with great pleasure that I hereby provide comments as respondent for Professor Mkhize-Kwitshana's inaugural lecture titled From Co-Infections to Cancer, Unraveling the Immune Thread of Neglect. I'm Gerhard Walsel, Distinguished Professor and Head of the Division of Immunology and Executive Head of the Department of Biomedical Sciences at Stellenbosch University. The lecture highlights how neglected tropical diseases modulate immune responses to HIV, TB, and cancer through immune suppression, skewed immune responses towards detrimental immune phenotypes, and altered immune checkpoints. The convergence of malnutrition, poverty, gut microbiome dysbiosis, and environmental exposures in the presence of co-infections could have detrimental effects on the control of HIV, TB, and malignancies. I congratulate Professor Mkhize-Kwitshana on the timely and thought-provoking lecture. The theme is highly relevant, particularly in the context of South Africa, but also global health inequities and global deprioritization of certain conditions, which led to the coining of the concept of neglected tropical diseases. The lecture bridges immunology, infectious diseases, cancer biology, and the effect of environmental exposures, a nexus often overlooked but clearly important. She covers several themes in the lecture. Co-infection as immune modulators. Neglected tropical diseases modulate immunity, worsen outcomes in HIV and TB and in cancer through immune suppression and inappropriate T helper 1 immune skewing, thereby undermining effective host defenses. Upregulation of immune checkpoints further constitute barriers to protective immunity. She covers intersection with social determinants of health. The compounded burden of malnutrition, poverty, gut dysbiosis, and environmental exposures underscores the importance of social determinants of health, culminating in the synergy of neglect, where biological vulnerability intersects with social economic disadvantage. She covers ontogenic pathways. I need to reinforce the point that chronic immune modulation by infections is not benign. It can pave the way for cancer. The global efforts in cancer research may not place enough emphasis on high-burden infectious diseases settings. Cancer research is often focused in well-developed, resource-rich regions of the world, whereas cancer also has a devastating impact on resource-scarce environment like ours. There are public health implications, integrated, simultaneous approaches that address neglected tropical diseases, HIV, TB, and cancer are needed, particularly in preventive healthcare, which has to be underpinned by appropriate research. She points out research frontiers that lend themselves to further exploration. Firstly, a potential fruitful research focus on underlying immune checkpoints due to neglected tropical diseases as a translational bridge to cancer immunotherapy. And secondly, an expanded focus on microbiome research in African contexts. Equity and neglect. Neglect operates at multiple levels, scientific, policy, and funding. And her lecture is a call for decolonized health research agendas that prioritize diseases and conditions relevant to the global south. In closing, I want to commend Professor Mkhize-Kwitshana for weaving together the immune thread across co-infections, social determinants, dysbiosis, and cancer. Her lecture is both a scientific contribution and a call to action for researchers, clinicians, and policymakers. Tackling co-infections, like with helminths, and their downstream effects is essential for advancing health equity. I congratulate Professor Mkhize-Kwitshana on her role, her position, her vision to weave seemingly diverse topics together into an integrated tapestry for the promotion of improved health for neglected parts of the world. I wish her well in her role as academic, mentor, teacher, and health activist.
Ladies and gentlemen, let us give Professor Mkhize-Kwitshana a round of applause for his inaugural lecture. As I said earlier on, if you are not inspired by this inaugural lecture, then you are expired. As for me and my house, when I grow up, I will definitely want to be like Professor Mkhize-Kwitshana and also deliver the inaugural lecture. Professor Mkhize-Kwitshana, welcome to the League of Extraordinary Gentlemen and Women. Siyaw amukela. You have asked relevant questions such as, are adults in South Africa affected by intestinal parasites? If so, what are the effects of these infections on other highly prevalent diseases? From the inaugural lecture, I learned that indeed adults in low-resourced, high transmissions areas are also infected, but neglected without targeted programs. Most neglected tropical disease, or what you called NTDs, are asymptomatic and overlap with many other high-priority diseases. One may also be tempted to ask, what then are the implications? We don't want to die because of lack of our knowledge. As Hosea 4 verse 6 says. I didn't hear any amen. In the absence of amen, let me be the first one to congratulate Professor Mkhize-Kwitshana and welcome her to the professorate. I've learned a lot today about some words that I cannot even pronounce, like helminth parasites, which are worms and are chronic and can live up to 25 years in the absence of treatment. And reinfection is very common, as we were told. So we want to thank Professor Mkhize-Kwitshana for being one of those who heeded the Tumamina call long time ago and be able to reach the pinnacle of academia by attaining professorate so that she can also contribute solutions to grand societal problems. For me, events like this inaugural lecture have the power to transform, the power to inspire, the power to motivate unlike any other. Ladies and gentlemen, we thank Professor Mkhize-Kwitshana for feeding us intellectually through the lecture. As for me, I am already fed up. Well fed by the lecture. I'm also thank the executive team of the College of Agriculture and Environmental Science, the respondent, Professor G. Walls from the University of Stellenbosch, family, friends, colleagues, and cousins. We thank you all for embracing the event, and we are saying, le ka moso ge le kwele mokgoši le re phalaleleng, and attend this inaugural lecture. Le lena ge le na le meletlo le re meme, re tla buša maoto ka gore bakoni ba re maoto a tla tswana. Siyabonga.